so let's fill in a few of these pieces here because we've already covered them. The tRNA molecule is a small RNA molecule with a clover leaf shape. The anticodon loop recognizes the mRNA codon. The amino acid attachment site is at the 3' end. Right? And attaches to an amino acid. Oh, and here it just tells us there are 45 different tRNA molecules. That's interesting. I thought there might be 64 tRNA molecules to correspond to the 64 codons. This is just interesting. We'll have to think about that in a minute. What does the tRNA do? It's the adapter molecule. It translates the nucleotide codon into an amino acid. Right? That's its job, to translate the codon into an amino acid. Now, a tRNA that is bonded to an amino acid is called a charged tRNA. And we'll see some pictures of this. Charging up a tRNA molecule requires an enzyme to put an amino acid onto our tRNA molecule. Remember, our tRNA molecule has this anticodon loop. To put an amino acid on, there's our amino acid, it takes an enzyme. It's also going to take some energy. The enzyme is in a group of enzymes called the aminoacyl tRNA synthetases. How many different aminoacyl tRNA synthetase enzymes are there? Well, there's 20 different amino acids. Guess how many enzymes there are? 20. 20 different enzymes to attach these 20 different amino acids to a tRNA molecule. Here's a picture of that. Here is our tRNA synthetase. Okay. It looks a little bit like an embryo, but what it's doing in this picture is it's grabbing an amino acid and a tRNA molecule. They enter the active site of the enzyme. This takes some energy. Attachment forms, and now we've got a charged up tRNA molecule. We call that a charged tRNA molecule. The tRNA molecule up here is uncharged. Right? And now we're ready to use this tRNA molecule to translate. It's ready to go. Okay. Oh, before we move to the next piece of our machinery for translation, we have a quick kind of funny little quirk to deal with. And this is called the wobble hypothesis. There are 45 different tRNA molecules, but 61 codons. So there are 45 different tRNA molecules. There are 64 codons, but 3 of them are stop codons. So there's 61 active codons that code for amino acids. Shouldn't there be 61 tRNA molecules to recognize each codon? This is weird. How can this be? We call it the wobble hypothesis. Some tRNA have anticodons that recognize 2 or more codons. In other words, we don't need 61 tRNA molecules because we have a little bit of overlap. Some tRNA molecules can recognize more than one codon. How does this work? Now let's read this and then we'll give you an example. The rules for base pairing remain the same for the first 2 bases of a codon, but the pairing rules for the 3rd base on the mRNA molecule are loose. There's some wobble in our base pairing. And let's show you an example of some of this kind of wobble, okay? So what I've written down here are 3 different codons: GGA, GGU, GGC. They're all running 5' to 3'. Each of these codons, if you go to that dictionary, codes for glycine. It turns out there's one tRNA molecule that can recognize all 3 of these codons, and its anticodon loop. So I'm going to write down anticodon loop in the tRNA molecule that can recognize all 3 of these runs something like this. It runs the opposite direction. Okay, so G, the first nucleotide in each codon is G. So what does G pair with? C. G pairs with C again. So the first two nucleotides in each codon pair with C. So our anticodon in this tRNA molecule, the anticodon loop goes C, C, and then you get to the third nucleotide, and the third nucleotide in each of these codons is different. What can pair with A, U, and C? Which nucleotide does this? It turns out that our tRNA molecule that can recognize all three of these codons has a funky nucleotide in it. It has something called inosine instead of an A or a T or a C or a G, or A or a U or a C or G. It has something called inosine. I know seen it before is what I always say. That's not fair. We didn't even know this existed. Apparently, inosine can pair with C, it can pair with U, it can pair with A. It can recognize all three of these codons. This one tRNA molecule can recognize all three of these codons because it has inosine as its third, as a part of its anticodon loop. Tricky, huh? And this would code for lysine. That's what happens with the wobble hypothesis. Okay, so now we know that there are these tRNA molecules that can recognize codons and bring in the appropriate amino acid. But we also need a place for this to happen. And if you remember from chapter 6, we learned that ribosomes are the protein synthesizing machines in the cytosol, right? Well, it turns out that we need ribosomes to synthesize these proteins. They're like the factory where this happens. So we need to look at their structure, and we need to look at how they can be involved in making our amino acid chain, or making our proteins, right? Okay, it turns out that ribosomes are pretty complicated. They're made up of proteins and ribosomal RNA. They're made up of two different subunits, a large subunit and a small subunit, made up of ribosomal RNA plus proteins. Okay, and this one's made up of ribosomal RNA plus proteins. So they're complicated. But let's take a look at specifically their structure. There are spots or binding locations in the ribosome, binding locations for the tRNA molecule, and binding locations for the mRNA molecule. They bring together the mRNA and the tRNA so that translation can happen. Okay? When we look at like these pictures of ribosomes, this is the kind of pictures that we get. Our textbook likes to use kind of these big brown blobs for ribosomes. Each ribosome has 3 binding sites for tRNA and 1 binding site for an mRNA molecule, kind of between the small and large subunit. The 3 binding sites for the tRNA molecules are called the E, the P, and the A sites. You don't have to know these exactly. These are places where the tRNA molecule can bind and recognize, when it recognizes the codon on the mRNA molecule. I want to show you I want to make a drawing of this. So I want to, I'm trying to figure out what I had in my notes here. I want to draw out kind of an example of how translation happens at these ribosomes. But just recognize that there are sort of 3 spots where our tRNA molecules can bind, the E, the P, and the A site. And let's review for just a minute. Oh, my dog is scratching at the door. Let me go get him a second. Come on. Here he comes. Okay, he just jumped on my bed, and he's trying to eat my food. I'll show you a picture of him. This is the dog that nearly died a couple of weeks ago. Oh, maybe you can see him better now. He's pretty healthy, but he's kind of scrappy looking because we cut his hair without a groomer, and it looks pretty bad. He's kind of naughty. I have my my home office here in my bedroom, so he's on my bed. He's trying to eat my snack. He can't eat any food, any people food ever again. So he's pretty hungry for snacks and such. In any case, He's just being naughty. I'll have to let him out in a couple seconds. No, he's making noises. Okay, let's just do a quick review for a minute. What we've got up here is a picture of a cell, and transcription is happening inside the nucleus. We had a gene that needs to be transcribed into an RNA molecule. The RNA molecule is not quite ready right away. It has to be processed. A cap, a tail has to be put on the mRNA molecule. The introns have to be cut out. And now we've got a processed or a mature mRNA molecule that leaves the nucleus. Once we're out in the cytoplasm, that's where translation is going to happen. Okay? That's where translation is going to happen. We need to have the mRNA molecule. We need to have tRNA molecules. Those are the little light blue ones that are charged up with amino acids that are purple. And we need to have the machinery for the factory where this can happen. That's the ribosomes. Okay? So how do the ribosomes, the tRNA, the RNA come together so that we can translate the mRNA molecule? That's what we're looking at, okay? Building a polypeptide in 3 steps. That's really what we want to figure out. How does this happen, all right? And the first step is initiation, getting started. What's going to happen first? So that's where we are, and that's where I'm going to start you. I'm going to draw this out. There's also diagrams here. Good. We can look at both. So we have this mRNA molecule. And so let's write that down, that this is an mRNA molecule. And we're out here in the cytoplasm. Okay? Right? And we have on this mRNA molecule nucleotides, right? At some point on the mRNA molecule, kind of close to the 5' end, is the nucleotide sequence AUG. That's our start codon, okay? And what happens when this mRNA molecule is translated, the first thing that happens is the small ribosomal subunit, binds to the mRNA molecule, all right? Then a tRNA molecule, we call it the initiator tRNA molecule, that recognizes the codon AUG, binds to our complex. So this tRNA molecule is going to bind to my mRNA molecule, and it has the anticodon that complements AUG. So it has the anticodon UAC. Oopsie, that's a C. Okay? It carries with it the amino acid that AUG codes for. So it carries with it methionine. Okay? Next, the large ribosomal subunit binds to our complex. Okay, and what we recognize is that as this large ribosomal subunit binds to the complex, remember there were 3 spots in the large ribosomal subunit where the tRNA could bind. There's an E site, a P site, and an A site. Okay, the initiator tRNA molecule has bound at the P site. Right. Next, that's how we initiate. The ribosome binds to the mRNA molecule such that we can start translating the mRNA molecule at the start codon, AUG. The tRNA comes in, that recognizes the start codon, it carries with it methionine. All right, we're ready. We've initiated this process. Okay. What happens next? Elongation. Okay, that's what this slide is showing. I'm going to draw a few more things on my slide here because I want to draw them in. So in elongation, the RNA, the ribosome then essentially has to read the next, or we have to read the next codon, UUU, has to be read and translated. And then the next codon, CCC, is read and translated. And then the next codon, and the next codon, and the next codon, right? We have to read, translate them, and then we need to put together the amino acids into a long chain as we translate them. That's what's happening at the ribosome. We're building proteins, building sequences of amino acids. So the first tRNA molecule is in place. It has read the codon AUG. It brings methionine. Next, we need a tRNA molecule that recognizes the codon UUU. So what anticodon will this tRNA molecule have? It will have AAA, all right? And it binds to the ribosome at the A site. Remember, these three sites are the E, the P, and the A site. And it has the anticodon A. What does the codon UUU code for? Which amino acid? Well, look it up in the dictionary. It codes for phenylalanine, PHE. All right, now we've got a problem. We have to connect our two amino acids to each other, right? We need to connect methionine to phenylalanine to get the first peptide bond between our two amino acids. And that's what happens next, okay? So let's switch over to this slide so we can look at this process of elongation, okay? We're kind of up here at this location. Right now we have one tRNA molecule with methionine attached at the P site. We bring in a second tRNA molecule that recognizes the codon at the A site, if you will. That one happened to be bringing in phenylalanine. And that takes some energy. Okay. Step 2, now we have to connect our amino acid that's connected to the tRNA in the P site to the amino acid that's connected to the tRNA in the A site. That process right there involves the moving, essentially, of the amino acid from the tRNA in the P site, shifting it over to the amino acid of the tRNA in the A site. And so now, if we look down here, a peptide bond forms. That's a covalent bond that connects our two amino acids like our poppet beads. And then we now have a tRNA molecule that's empty, right? It doesn't have any amino acid on it anymore. And we have a tRNA molecule in the A site that is all charged up with this nice chain, okay? And this one happens to be longer than ours. But what has to happen next is there has to be a translocation or a shifting so that we can get the tRNA that's in the A site shifted over to the P site, and we can open up the next codon to be read, in other words, to be recognized, and a new tRNA can come in and bring in its appropriate amino acid. And this goes on and on and on. What happens to this tRNA that's uncharged? Whatever happens to that? Well, it enters the exit site or the E site temporarily, and then it leaves uncharged. It doesn't have any amino acid anymore. It has to be charged up if it wants to be used again in translation. But our growing chain of amino acids sticks together, and as we translate more and more and more, we get a bigger and bigger and bigger chain of amino acids. So in my drawing, I would need to think about how this ribosome and the mRNA molecules are kind of shifting past each other, sliding by, so that we can always open up a new codon to be read. So at this point, what would have to happen is methionine would have to connect to, form a peptide bond to phenylalanine. This would be methionine is my first amino acid, phenylalanine is my second amino acid. Then our tRNA molecules would have to shift, I don't know why that is, would have to shift one position over, okay? Each one of them would have to shift one position. And then we could read the next codon. The next codon would sit in the A site and be able to be read. The next codon is CCC. The anticodon, the tRNA's anticodon loop that recognizes CCC would be GGG, and it would be bringing along, I have no idea what CCC codes for. I'd have to look that up. You guys could maybe look up CCC. Oh, maybe it codes for, I have it in my notes. It codes for proline. So this is our next tRNA that's going to be entering the ribosome at the A site, and we're going to build a longer amino acid chain. And we're going to keep doing this until when? We're going to keep doing this. Notice that there's energy used in this process, and that's no surprise. We're building a protein. That's going to be energetically expensive, right? When does it all stop? Like, when does it end? When does translation get terminated? Okay, that's the question I ask. So does translation get terminated? Do we just kind of keep translating until we run off the end of the mRNA molecule? Or do you remember something about stop codons? When the translation machinery gets to a stop codon, like let's just use UAA, that's when translation ends. Okay? The termination of translation occurs when you arrive at a stop codon. So so the mRNA is being translated, the ribosome and the tRNA molecules are translating it until you arrive at a stop codon. There are 3 stop codons, UAA and UAG or UGA. When you arrive at one of these, then translation stops. There are release factors, shown here as little yellow blobs on my diagram. Release factors that bind to a stop codon. And when that release factor binds to a stop codon, translation ends. All of the pieces of translation fly apart, basically. So tRNA molecule leaves. There it is, the free polypeptide. The amino acid sequence that we just made is free. The mRNA is released. The ribosomes are released. The ribosomal subunits come apart. All of these parts can be recycled. They can be reused, right? And the polypeptide, the chain of amino acids, will be, maybe has to be modified. Maybe it has to be folded properly. Maybe it has to be put into a bigger protein. But eventually it will become a functioning protein. Right? There's a nice animation of translation that you can watch that might be useful for you. A couple other components here. What we do notice is that during translation, many ribosomes can be working at the same time to translate an mRNA molecule. And so here's a nice picture of an mRNA molecule with lots of ribosomes on it, doing the job of translating. Those little blobs there are those tiny little ribosomes doing the job of translating the mRNA molecule. We call that a situation of polyribosomes. Okay? That just makes translation faster, right? And I already mentioned this, after translation occurs, we get this cute little chain of amino acids, but that chain of amino acids might have to be modified. It might have to be folded. It might have to be added together with another polypeptide chain to make a complex protein. Lots of happening still occurs. And we might have this happening in the rough endoplasmic reticulum or in the Golgi apparatus, right? Those are places where modifications of our proteins occur. Whew. Back to chapter 6 material, right? So we've just translated. I think we're near to the end. This is a nice summary sheet that if you can explain this, you can say, oh, okay, what's happening in transcription, mRNA processing, what's happening in translation. This will be helpful to you as you're thinking about the next online quiz. There's also a nice animation here that is worth looking at. So we've covered translation. I'm going to quit now and we'll come back to part 3.